don't worry, I'm not eating this time. But I do want to talk about the proteins. Last time we talked about carbohydrates. This time I'm going to talk about the proteins that I actually ingested as well, and the enzymes, and what exactly the enzymes are doing and how they're doing their job inside of my digestive system right now. So let's dive right in. First of all, proteins. What are their functions? Well, major function is that they can help build the basic structures and materials for the cell and the cell parts. Proteins are also, they can build enzymes, all right, and enzymes are actually proteins, so proteins can build proteins. And proteins can also, they're used to grab onto the oxygen and the hemoglobin in the blood, because oxygen isn't very soluble when it goes from gas to liquid, so it's able to grab onto it and help it make it a little more soluble for our body. And then if you look inside of our muscles, they are just packed loaded with protein, right? So when you get done with a big workout, that's why a lot of people go for protein shakes. They want to build up those muscle cells even more, and that's where the protein comes in. We're going to get more into muscles and protein in another unit during this year. So here's a vocab check real quick. So we got proteins, and PEP, P-E-P, that means protein. So we have polypeptides, we have dipeptides. And so polypeptides, just like poly meant before the saccharides, means many. So many protein monomers linked together. And then dipeptides, di meaning two again, so two protein monomers as well. But kind of just like the carbohydrates, they can break through hydrolysis as well. And that's actually a key important facet to remember as we move on. Let's get a quick overview here of the protein digestion that's going on. So the protein in my body right now from that pizza, that glorious Papa John's pepperoni and green pepper, that pepperoni, all right, let's think about that pepperoni that's going on here. It's being digested in my stomach using the enzyme pepsin. All right, so there's a new enzyme for you. Last time we were talking about salivary amylase, now we're talking about pepsin. Now, from there, it's going from a polypeptide, the big protein down into the polypeptides, right? And then that's then digested even more in the small intestine with the enzyme trypsin and protease. And that's going to break it down even more into dipeptides. And the dipeptides can then be absorbed into the bloodstream. So enzymes have that globular look to them. I talked before about how it's kind of swirly, the strands are swirly, and they're folded over, and their structure, the way they're folded, gives them their function. How? Well, because their structure will allow specific items called substrates to fit in there with them. Kind of think of it as like a lock and key, where the lock is the enzyme and the key is the substrate. So not all locks are made for all keys. And they're different, and you see here in our examples here, there are a lot of different ways that this could, this lock mechanism could be made. And this is just a few of them here, all right? So they have one specific structure, so they have one type of reaction or one specific substrate that they can work with. That's why we have so many different enzymes. Now the enzyme, like I said before, is going to act as a catalyst. The catalyst speeds up a chemical reaction by lowering the amount of activation energy that's needed to conduct the reaction. So if we take a look at our model here, model A here, there is a big hill that this ball must get up before it can start its roll down. And from here on down, that would be the reaction. So this is just a buildup right here. So a lot of chemical reactions, before they can actually occur, they have to build up a lot of energy before that can happen. And in this case, that's what's happening. Now, over in Model B, we can see that slope isn't as big. And that's where the enzyme comes in. The enzyme lowers that slope, so you don't need as much energy for that chemical reaction to begin. So then, if you don't need as much energy, you don't have to wait around as long to build up energy, so then the reaction can happen quicker. And the enzyme also helps by taking substrates and bringing them together. All right, that's another big way that it's going to help speed up chemical reactions instead of waiting for random bumping into of different chemicals, molecules for a reaction to occur, and at the right angle and at the right amount of energy or speed behind them as well. So here's a kind of a overview of an enzyme at work. We have an enzyme, we have an active site, right? The active site is where the substrates are going to plug into. In this case, we have two amino acids, and these amino acids at the end are going to be a dipeptide. All right, so let's take a look here. So we have our enzyme. The 2 amino acids will lock in to their, so again, these are the keys, and here's your lock. So I'm going to go in there. They fit just great. And when they do that, we now have what's called an enzyme-substrate complex. Enzymes and substrate together. Now, from there, we're actually going to remove water. Remember last time when we talked about the carbs, salivary amylase, and breaking down saccharides into the disaccharides into the monosaccharides, how we use a hydrolysis reaction. We're going to use a dehydration reaction here. And we're going to pull that water out, okay, which is then going to cause these two to bond together. And then from there, the enzyme will release a substrate, and now we have a dipeptide product with a peptide bond there. Now, with that removal of water, these two chemical molecules here, they're going to bond together so that they can share the right amount of electrons so that they can become stable, so their valence electrons can be stable. Remember chemistry class? I'm sure you do. You better. So the removal of the water takes electrons with it that were being shared before, and so that's going to help out there. Now, enzymes, they need vitamins a lot of times, right? So their activity is improved by the use of vitamins. In fact, enzymes are sometimes with a vitamin. The vitamin is called a cofactor, right? So the enzyme isn't active without its cofactor, the vitamin. So the vitamin attaches to the enzyme, and now the enzyme can do its job. And a lot of times, the cofactor, the vitamin, it actually has the binding sites for the substrates on it. So the protein couldn't do anything anyways because it didn't have any binding sites. So now it does, and you can see now that our substrates can now connect to our enzyme. Now we can slow enzyme activity, or we can stop it using what's known as inhibitors. So in this case, here's our substrate that is actually what this protein, this enzyme, was made for. You can see it's going to fit in there quite well. But this competitive inhibitor actually goes on in and actually does not allow this substrate to go in there to the active binding site. So whatever reaction this was has been inhibited there. Now we can also do what's known as a non-competitive inhibition. And in this case, it looks like the substrate can fit in there just well. There's no competition there. There's nothing going in there. But this non-competitive inhibitor actually goes in another part of the enzyme and actually then changes. It pushes around the coils of those strands of the protein and then causes it to change its shape. And now again, since the shape has changed, the function has changed. In this case, it can no longer function for that substrate. So that's another way that enzyme activity has been slowed or stopped by that non-competitive inhibitor there. Now we can also denature enzymes. Denaturing enzymes comes with both temperature and pressure. We can heat up, we can drop the temperature, we can raise or lower the pH as well, and that's going to affect the form of that protein. So remember that globular, coiled up form of all those strands. Well, when we denature them, we can actually cause a random coiling. We can change the shape of it that way. Now, if it was only denatured just a little bit, so the temperature is just changed just a little bit, the pH just a little bit, the protein can actually, the enzyme can actually go back to its native form. But if it's for a long amount of time or the temperature is so great, it's going to change it, right? Actually, when you're cooking meat, you're denaturing the proteins of that meat. All right, so that's a quick overview of proteins and enzymes. If you have any questions, bring them to class.